Today we are going to talk about vaccination or immunoprophylaxis, which is prevention of diseases by vaccine. But let us first know the source of the word vaccine. Vaccine, it comes from a Latinic word vacca, and vacca, it means cow. And this was on the honor of the British physician Edward Jenner, who worked with cows during the 17th century. And as you know that vaccines are either natural or artificial. Natural means you do not seek service. You do not go to the hospital to get vaccine, but you get it naturally. It is either passive or active. Passive means that, for example, during pregnancy, antibodies will pass from the mother to the baby, so to the fetus. So the fetus will get antibodies, usually of IgG, from the mother in a passive work. And these antibodies will protect the fetus in utero and will remain in his body for 6 months following delivery and protect him during this period. So we call it natural passive vaccination. A natural active vaccination, it, for example, or the example of this vaccination, if someone get bitten by a mosquito, the mosquito will inoculate sporozoites in his body. This is actually the natural part. But the active part, now the body will see these sporozoites and start to upregulate an immune response against them. So this is, we call it the active part of the natural active vaccination. So in artificial vaccination, usually an individual seeks service, and it is also divided into passive and active vaccination. So artificial passive vaccination, the example for it is injection by ready-made antibodies. For example, protection against hepatitis A. So these antibodies are not formed in your body, but they arrest outside the body by many means. And also in case of a tetanus patient, they get anti-serum containing antibodies against the toxins. So this, we call it artificial passive vaccination. So artificial active vaccination, it means that the body is injected by either live attenuated vaccine. Here the microorganism get attenuated by many means, and then deliver to the body. And when the immune system sees these attenuated organisms, start to deal with them. For example, the macrophages will take these microorganisms, will process them, and as can be seen here in this figure, this is the vaccine, will be taken by the macrophage. This is the attenuated, life attenuated microorganism or vaccine will be taken by this macrophage in green, and then the macrophage will process the microorganism into peptides as seen by red dots, and these peptides, they bind to major histocompatibility class 2 at the rough endoplasmic reticulum. So peptides derived from this weakened or attenuated live vaccine together with the major histocompatibility will be expressed on the surface of the macrophage. Now T helper 1 will see peptides derived from the live attenuated microorganism or vaccine, and through the T cell receptor it interacts with these peptides, and through a protein on their wall known as CD4 will interact with a major histocompatibility. This will lead to activation of the T helper 1. Activated T helper 1 will produce interleukin number 2, and this interleukin number 2 in turn will activate T helper 2 and B lymphocytes. And as you know that B lymphocytes are antigen presenting cells, and also they take this attenuated vaccine and process it and present it to T helper number 2. So T helper number 2 and B lymphocytes are activated by the interleukin number 2 from T helper number 1. And this result, the interaction between B lymphocytes and T helper 2 result in activation of B cell. The activated B cell will differentiate into memory B cell, and plasma cell. The plasma cell will produce antibodies, and these antibodies in turn will protect if the individual challenge with the microorganism in the future, and memory B cells will remain, and if they encounter the microorganism in the future, they will give antibodies. And also, one of the advantages of the live attenuated vaccine is not only formation of antibodies, but also stimulate cellular immune response. And as you know that in case of cellular immune response, here T helper number 1 will produce interferon gamma, and this interferon will recruit more macrophages to the area. So the recruited macrophages will get activated, and they will produce cytokines, pro-inflammatory cytokines known as TNF alpha and interleukin number 1 and interleukin number 12. Interleukin number 12 activate more T helper 1 cells. Activated T helper 1 cells will produce more interferon, and more interferon will recruit more macrophage. So this we call it cellular immune response. So in case of live attenuated vaccine, both humoral and cellular immune responses get activated. But however, in case of killed or inactivated vaccine, like in flu and rabies, the killed or inactivated vaccines, they simulate only antibody immune response. So here the individual only mount or upregulate or produce antibodies, but however no cellular immune response. The example of live attenuated vaccine is BCG, which is Bacillus calmette-guerin, which protects against tuberculosis, and it is weakened or attenuated Mycobacterium bovis. And also one of the examples of artificial active vaccination is toxoid, like in case of tetanus. So here, toxins, they are not injected directly for the purpose of vaccination, but they are processed pharmacologically to remove the toxicity. Recently, one of the most recent vaccines are recombinant vaccines, and this depends on this picture. Recombinant means that the vaccine should contain some epitopes. Epitopes, it is an immunologically active part of the antigen, and these epitopes should be recognized by B cell and should be recognized by T helper cell in order to produce antibody immune response.